Ladies and gentlemen, welcome back to the continuation of our session. Thanks for taking a seat. So we are continuing with our session, and I'm very pleased to introduce the next speaker, which is Dr. Jolyon Medlock. Dr. Medlock has been working on vector-borne diseases for 20 years, and he was trained at the Universities of Manchester, Bristol, and Liverpool School of Tropical Medicine. And since 2002, he has worked at Porton Down for the Health Protection Agency, now Public Health England. Dr. Medlock leads the medical entomology department, and he also frequently advised the UK government on vector-borne disease. He's also been very active at the international level, and he will introduce us his talk on vector-borne diseases in Europe, what can we expect, and how well are we prepared. Dr. Medlock, the floor is yours.
So now for something completely different to what we've heard already, and I'd just like to say thank you very much for inviting me. I sort of come here as a medical entomologist working within public health government, but also have spent the last 10 years working on ECDC and EFSA funded projects on vectors as well across Europe. So you might be familiar with one of these pests that I'm going to talk about because they've been biting in the eat and meat area, eat, meat and bite. So I've already killed a few of these mosquitoes. But I think when I started my career as a medical entomologist, focus pretty much was on the tropics. And actually these vectors and these pathogens only really occurred in tropical areas, which is all very exciting. But actually over the last 18 years of this century, vector-borne diseases have re-emerged as an issue in Europe. And here you can see some of the examples. I'm going to talk a bit about exotic mosquitoes and the way in which they've managed to colonize Europe and become involved in the transmission of tropical diseases. We've also seen the emergence of West Nile virus, not only in North America, but also in Europe, and particularly this year. We've had Lyme for quite a while, but it's on the increase. We've also had the movement of other pathogens and the incursion of things like blue tongue virus in the veterinary side. The expansion of tick-borne encephalitis up to Scandinavia. Then in the second decade, it carries on. We've had malaria outbreaks in Greece. We've seen dengue transmission in Europe for the first time in nearly 90 years. We've had the emergence of new pathogens, and of course Zika really was one pathogen that actually alerted the world to potential threats of a novel virus causing unpleasant consequences. So a vector is any agent, I should say, that carries and transmits an infectious disease. So I'm talking about arthropods really. Mosquitoes and ticks are the things I work on mainly, but also sandflies, fleas, midges, and sometimes rodents. Now, I think 75% of emerging infectious diseases are wildlife zoonoses, so they're pathogens that occur within wildlife and also transmitted to humans. So there's never a greater advert for the need for One Health. Now, this here illustrates our One Health policy in the UK, where on a monthly basis, actually since BSc, it was decided that we need to have a proactive approach to understanding the next threat. And this is led at the top with the Chief Medical Officer and Chief Veterinary Officer and has representatives both from public health, clinicians, veterinarians, but then as also we're dealing with things like vector-borne diseases and also pathogens, we need virologists, bacteriologists, entomologists, and also the expert group that work on the particular livestock areas. So for the last 14 years or so, we've met on a monthly basis to try and assess what the risks might be. Now, these are just an example of 20 questions that I might get asked on a monthly basis. And you generally are expected to have the answer there and then at these meetings. So it necessitates a need to plan your surveillance and research in advance so that actually you're the one raising the issue and you're also, when they ask for the answer, you're able to give it. But these things are not just entomology. You need to have an understanding of ornithology, of wildlife ecology, habitat management, environmental change. I mean, one of the questions at the end there is, what effect will heat waves, floods, coastal flooding, climate change have? Well, you could probably write several thesis chapters on that one. The end result, actually, is these are the questions that the public want to know. They want to know quite rapidly. Particularly in the case of Zika, we had 5 questions from a government minister, and we had a couple of days to answer them. And obviously, with a novel virus, that requires an awful lot of information gathering that you don't really have time. So you need to be thinking ahead. And if I say once, I'll say many a time, you need to have continuity and sustainability of surveillance and research that's targeted towards policy questions, because you can't make this up as you go along, because you'll get found out, particularly as a government scientist. Now, our work ends up leading to these, the Human Animal Infections Risk Surveillance Group is this One Health group, and these are some of these risk assessments on Zika, or newly emerging tick-borne bacteria, chikungunya, and that then is publicly accessible on our government website, so any academic, any member of the public can interrogate it, so it needs to be pretty accurate and updated on a regular basis. So I'm going to start off talking about invasive mosquitoes. Some of you will be familiar with these. We currently have 4 invasive mosquitoes that have colonized Europe in the last 30 years and are expanding rapidly. I'm going to start off with Aedes albopictus, known as the Asian tiger mosquito. It's very much a European mosquito now. This mosquito is a tree-hole mosquito of Southeast Asia and has had the ability to move around the world by laying its eggs, drought-resistant eggs, in products that we move around for it. So things like used tyres or lucky bamboo. It was reported in Albania in the late 70s, but really came to prominence in the early 90s in Italy, where it subsequently has spread to large parts of the country, even to these buildings here. It doesn't actually fly very far itself, only a few hundred meters, but it relies upon us to move it. And it's then been spreading along highway systems down the Mediterranean coast, as far as Gibraltar and Algeciras, now from Italy, and also down the Croatian coast, meeting up with its population in Albania. And the latest map shows actually quite a wide distribution in southern France. And I'll mention briefly, we've detected it 3 times now in southern England. So it has a pretty efficient way of moving long distances. ECDC have been very supportive of the work on Aedes albopictus over the years, and actually funded some risk mapping almost 10 years ago now, and this is one of the products of that risk map, which doesn't look much different to the distribution map of albopictus 10 years later. And a number of countries, including my own, have produced models looking at climate suitability and abundance models, and also using climate change ensemble data. Others have taken that further forward by looking at the pathogens that might be transmitted by the mosquito. If you think about a virus having an extrinsic incubation period within the mosquito, being temperature dependent, you can then look to see whether we will have transmission of these viruses in the future. Now, I remember sort of 10, 12 years ago being really told to stop scaremongering about the prospect of albopictus and chikungunya in Europe, but I think, as history's shown, that actually often what you think might happen as the scientist generally does see through, and we have had outbreaks of chikungunya since. There are a couple of other mosquitoes worth mentioning. Aedes aegypti, the yellow fever mosquito, was common right across the Mediterranean and actually colonized for the first time Madeira from about 2007 and is now re-establishing itself on the Black Sea coast. In time, it may well spread to other parts of the Mediterranean. Aedes japonicus, which is the mosquito in the middle there, was actually discovered whilst looking for Aedes albopictus, and that now is spreading right through Switzerland, and also up into Germany and the Netherlands. Potentially it could transmit West Nile virus or Japanese encephalitis, but it's not known to be involved in transmission in Europe at the moment. Now, it's all very well, so okay, we've got these mosquitoes, are we really going to see anything? Now, what happened in about 2005 was a mutation in the gene of one of the chikungunya viruses in La Réunion, which meant that it could actually be transmitted better by Aedes albopictus, and that led to 250,000 cases in La Réunion, 1 million cases in India, and subsequently, as some of you will know, an outbreak in northern Italy. Since then, we had movement of chikungunya to the Caribbean. This all happened really during the Ebola outbreak, so it was largely ignored by a lot of public health agencies, but over 1.5 million cases right across the Caribbean, with an index case in St. Martin in the Caribbean nations spreading to South America. And of course, Zika did exactly the same, but from a slightly different starting point. Now, timeline of European issues. As which you can see here really started in 2007. Since then, we've had the occasional case of dengue and chikungunya transmitted by Aedes albopictus. There was then 2,000 cases of dengue in Madeira transmitted by Aedes aegypti, and most recently last year, a larger outbreak in Italy. Now, these numbers are small compared to what you might find in other parts of the world, but it just shows there's an increasing trend as the mosquito becomes more abundant and we continue to travel, then there'll be more outbreaks in the future. Over the years, we have conducted a range of reviews, understanding the ecology, biology, disease transmission. There's guidance on surveillance. And I just want to mention VectorNet, supported by EFSA and ECDC. And this is a huge amount of work to begin to map these mosquitoes. Actually, when Zika was occurring in South America, from my perspective, it was where are the maps? Where are these mosquitoes in Europe? And actually, within a click, they could find these maps on the ECDC website, EFSA website. And actually, a lot of the work had been done for them. Of course, this needs to be carried on. This is a dynamic situation. People are finding these mosquitoes in new areas all the time. This is crucial for risk assessment. I was able to point to a website and say, Aedes aegypti is only in Madeira and the Black Sea coast. Aedes albopictus is only in these parts of France. We do not have it in the UK. And we probably saved ourselves an awful lot of money conducting extra surveillance and also doing alarmist responses. And of course, we could point the press to it as well, which was comforting. We do have to be prepared now for these tropical viruses in Europe. And one of the issues is not only surveillance, but actually how do we control the mosquito? And I just want to mention this new cost action called AIM, Aedes invasive mosquitoes, that's just been set up, which is looking to be homogeneous in our approaches to surveillance and also control. This is going to be a big task. And with these mosquitoes, it's not up to the government or the municipality to deal with it, it's everyone's responsibility. Everyone is harbouring these mosquitoes in their backyards if they're living in the areas where the mosquitoes occur. From a UK perspective, we now have surveillance at 37 UK ports. Probably a bit overkill, but actually people at port health officers are interested. They want to be sure that they're not harbouring any mosquitoes. Our biggest concern is mosquitoes coming in along highway systems through Eurotunnel as the mosquitoes spread through northern France. This actually is quite challenging because you have to engage with an awful lot of new people you wouldn't normally talk to. People who run lorry parks, environmental health officers, members of the public. It takes time. And of course, you haven't got time because the press like to write about Zika eggs found in southern England. So you have to have answers and you have to have everybody briefed. So for the UK, we've actually found Aedes albopictus 3 times now. And it's a real challenge to get up to speed and all local public health up to speed. We have quite a lot of people who come back infected with chikungunya and dengue every year. If we find that these mosquitoes are established locally, then they present a concern for local transmission, which clearly has been the case in France. So what are we going to do about controlling it? Did government do enough to eradicate the mosquito in the first place? I suspect this mosquito will establish in UK as it's established elsewhere, and then it'll be a case of minimizing outbreaks. And also, we need to make sure that the people who are responsible in the field, who actually have to deal with this on a day-to-day basis, who aren't medical entomologists working in public health England, have all the necessary information available to them to control the mosquitoes. Moving on to West Nile virus, this became prominent in the late 90s, starting in New York, and we all witnessed an outbreak that spread right across North America over the following 10 to 12 years, almost affecting nearly every state. You can see there the cases spreading really in a band as migratory birds move up and down, and also there were populations of mosquitoes that were suddenly susceptible to West Nile virus and able to transmit it. So from a disease ecologist point of view, it was a fascinating subject to study during those early years, but obviously caused an awful lot of cases and deaths as well. Now we have West Nile virus in Europe. Normally we would have between 200, maybe an extreme 700 cases. I got these data from ECDC yesterday, so I hope they're correct. But we have maybe 1,400 cases so far this year in Europe with 90 deaths. And you can see the larger outbreaks there in Italy, Serbia and Greece. This is unprecedented, I think, really, this year for West Nile virus. We haven't seen this sort of outbreak before. We can ask questions why that might be, maybe something to do with the climate. But I think West Nile virus isn't going to go away. And whether we see something quite as dramatic as we've seen in North America, only time will tell. So, from my perspective, what about the UK? Are we at risk of West Nile virus? Do we have these mosquitoes, and we're looking not Aedes mosquitoes, but human-biting Culex mosquitoes, and ones that have been bitten a bird as well as biting a human. So actually, Culex modestus is one of the key vectors in Europe. And prior to 2010, we didn't conduct any surveillance in the UK, so we'd have absolutely no idea whether we had this mosquito at all. And it's not the sort of thing you can get academics to do as part of research, because it's surveillance. What we did find actually in 2010 is that the population of potential West Nile virus vector occurred on the Thames estuary, and since then we've been monitoring its spread right up towards London and all the way up the coast. And it seems to be dominating. Some of these mosquitoes that are invasive are pretty effective at colonizing new environments. What we do now is we use this data to link in with the UK Clinical Virology Network, so anybody presenting with viral encephalitis in the area where this mosquito occurs is tested for West Nile virus. Otherwise, what's the point of collecting data on the mosquito if you don't then link it back to disease transmission, potential disease transmission? This map is updated on our government website, and I think it's really important that assessments are dynamic, and you need that surveillance data to link into it, and also that you're as transparent as possible with the public and also with clinicians. One element I just want to mention is about environmental change. I got asked, what's the impact of a 2, 4, and 6 degree temperature change on mosquito-borne disease? Well, actually, if you change the temperature that much, most of the coasts will be flooded anyway, and there'll be a lot more wetlands. So you have to think about wetland management, wetland creation, and we do an awful lot of that in an island nation. So actually, are we building or are we constructing new wetlands? Are we designing wetlands to make them disease-proof in the future? Is in the environmental impact assessment, is there a factor in there that says, have you considered future vector-borne disease risk? I don't believe there is, and maybe we should think about it. And actually, some of the opposition towards these wetland creations are down to them being mosquito-infested sites that may have future disease risks. And a lot of the accusations are false, but there isn't always the evidence to prove that. I'm going to just move on to ticks, conscious of time. The main tick-borne disease in Europe is Lyme borreliosis. There's an estimate of over 40,000 Lyme cases. Borrelia burgdorferi, the bacteria, has a very complex ecology. It can be found in a range of different animals. There are different genus species circulating between ticks and mammals, ticks and birds. The tick is very sensitive to changes in climate, changes in habitat, which actually makes an assessment of its risk quite difficult. Also, when you look in ticks, you find a range of other pathogens, and I think I'm not an expert in pathogen detection, but there's a huge scope to look and see what other pathogens are in ticks, and then linking those potential pathogens to possible disease. If you're negative for Lyme, what else might you have? And I just list a few there. Miyamoto was spelt wrong, I'm sorry, but... we've been conducting surveillance for ticks since 2005. We've now developed a longitudinal data set. That enables us to do a number of things. Firstly, to plot where Ixodes ricinatus is with the Lyme vector. We can then identify areas of the country where they're only recently being found. We can talk directly to members of the public. We can look at change in distribution, and also we can link it to risk mapping. We've been doing this at a European level. Just to mention VectorNet, I led a tick consortium for 3 years under VectorNet, and we provided building capacity for surveillance and monitoring in 30 European countries. And now we've got some European models, and we've been able to actually, for the first time, detect ticks in Iceland, where they're now beginning to develop a Lyme program. Also in parts of the Mediterranean, where they're concerned about hiloma. And also in parts of the Caucasus, where there's Crimean Congo hemorrhagic fever virus. And actually, this is one of the most rewarding projects I've been involved in, because actually you build a network of friends, you can share capabilities, you can share information, and when a crisis comes along, you can actually pick up the phone or email someone and share information. And I think actually we're better prepared as a result of this. Sustaining this network going forward obviously requires funding, and I think that definitely needs to be a focus, and I know it is a focus of EFSA and ECDC. Ticks aren't always just found in the countryside. They're increasingly becoming a problem in urban areas as well. This is where I live nearby in Salisbury, which is famous for something else other than ticks at the moment. And as you can see here, the ticks are in urban areas, and actually you get quite high prevalence rates, up to 30%, largely because there are no large animals around to dilute the transmission system. So there are potential conflicts between building in urban green space, encouraging people to use it, managing it for biodiversity, and the consequent effects of those animals moving in, bringing bacteria and causing disease. Using this information, you can then raise awareness, because actually it's what you have to do with tick-borne disease is encourage everybody to check themselves for ticks, and if they present with any signs, make sure they go to the doctor. And actually, without information, people can't self-prepare themselves. What we've also been doing is looking at monitoring tick activity over time as well. And this is a weekly transect that we do to see how ticks change in their activity throughout the year and over the years. And also, not only temporally, but spatially. So actually, if we've got long-term monitoring of tick density, this enables us to compare tick density by habitat. We can then monitor Borrelia prevalence within the ticks, and then we can calculate the density of infected ticks by habitat by year. So instead of just saying there's a lot of ticks there might not necessarily be many infected with Borrelia. So actually we can look at the density of infected nymphs. So this is the sort of surveillance that we probably ought to be looking at in building on the distribution data. Just to finish on a couple of non-native tick species, Rhizocephalus sanguineus, the dog kennel tick. We now have these ticks coming into the UK. I know they're common in the Mediterranean, but in the UK they're coming in on travelling dogs, and actually are now, instead of, they can't survive outdoors, but they can survive in people's houses, so they live in people's furniture, behind their wallpaper, and obviously creates a bit of a concern. So I'm making sure that we monitor ticks on animals moving between borders is quite important. Of course, we can't always do that, because migratory birds will move ticks themselves, and the vector of Crimean Congo hemorrhagic fever virus is moved around by migratory birds. In fact, this last week, we found a Hyaloma rufipes on a horse that hadn't travelled in the UK, and it had probably come in as a nymph on a bird, fallen off, molted in very warm summer weather, and now biting locally in England. So this is, just shows really with a very warm summer, anything's possible. CCHF is a very complex transmission cycle, and actually you have to have assessed the risk well in advance before suddenly somebody dies in Spain with CCHF. And I think this is very similar to Ebola in its clinical aspects, and something that we ought to consider for the future in Western Europe. So just to finish really, vector-borne disease incidence and distribution has changed in the last decade. I hope you agree with that. And now we've got to consider tropical vectors and tropical viruses to occur in Europe. Climate and environmental change. There's got to be a focus on linking in with biodiversity. This morning I sat in the environment session, talk about ecosystem services, and actually when we're changing the environment, are we considering what human health impacts? They're not all negative, but it's just make sure that we're considering them. Maintaining vector surveillance even in peacetime is crucial, because when a crisis comes along, if we're having to quickly make things up on the spot, we're going to end up wasting money, making the wrong decision. Vector-borne diseases tend to be zoonotic, so we need to work in a One Health, and actually it's not just medics and vets, entomologists have something to say as well. And of course enhanced detection. I think that's the key here is that there are other pathogens out there that we're not currently aware of that may be causing disease. So thank you very much again for inviting me.
Thank you very much, Dr. Medlock, for this interesting and very illustrative presentation. Are there any questions from the audience? Yes?
Hi, Jordan. Thank you very much for this nice talk. I'm Miguel Miranda from University of the Balearic Islands. I'm an entomologist working as well in the panel of animal health here in EFSA. I wanted just to comment on what you said on your conclusions. That is very important. Do you hear me okay? Or?
I can hear you.
Yeah. It's very important to keep vector surveillance in the country level in order to be prepared, in order to assess any possible threat for vector-borne diseases. But at what level do you think we should keep this surveillance? Because it's always the question with mosquitoes and so on, that we have the national level and the local level. And sometimes it could be very clear at national level, but not so clear with the local governments, at least in the case of countries like Spain. This is my first comment. How do you see how to implement this from a national to a local level? And secondly, you mentioned the Vector-net project. And I'm also curious to know your opinion about how do you think this Vector-net project, which is already more than 8 years working, is adding value to the risk assessment, again, at national level and at European level? Thank you.
Yeah. I think vector surveillance largely depends on the vector involved. And we all recognize that resources are limited and getting more limited. And actually, we need to think about how we can plug into existing networks for vector surveillance. So the tick surveillance that we do in the UK, it's a passive scheme. It requires people sending ticks to us from vets, from clinicians, and actually that gives us our information on unusual species, which then often lead to local investigations with the local authority. Obviously, you can't do that necessarily with mosquitoes, because they're not always as easy to collect as ticks, and they tend to get damaged, so you need to be more proactive. And you maybe need a network of traps, which becomes problematic in the fact that you need people to go and run those traps and identify the mosquitoes. Finding ways in which we can work with other groups that are monitoring species, such as entomologists for plant health or entomologists for biodiversity, who may already be actually servicing traps for longhorn beetles or something, and they can run a trap for you, or nature reserve wardens. A lot of our traps are actually run voluntarily by environmental health or nature reserve wardens, because we do not have the resources. What we do have resources for is to coordinate those traps and identify the mosquitoes. The challenge then with invasive mosquitoes is that they're in urban areas and they're quite cloistered in where they are. They're hard to find. And I think the only way in which you can deal with that is to actually start training people at a local level. Because if you've got the situation whereby you've got a big chikungunya outbreak somewhere in the world and lots of people are coming back infected, is there going to be any local transmission? You want to get in there as early as possible to minimize any biting risk and any local transmission. So I think really that has to be done at a local level, and people at a local level need to be trained to do their own surveillance and do their own control. And then if in the absence of national funding, it needs to be a local priority. Your other question about Vector-net, what is it used for? I think the utility of Vector-net data largely depends upon how well engaged the national government is with using that data. I actually find that I'm an entomologist working in Vector-net, but also within government, so it's crucial for me that those maps are available. But maybe we can, in the next project, make sure that we're sort of encouraging governments to utilize those maps better. Perhaps they have systems in place where they're monitoring vectors and input a bit more, and some case studies to show actually how they can be used, and in turn how they can probably save money. Because I do think that this is crucial to maintain this sort of data, because often I know people in EFSA and ECDC have to deal with things, the next crisis, and they don't have the luxury of building in time. So they've got people gathering that data through these networks to then inform when the crisis occurs. That's going to be the main way forward.
Thank you. Any other question from the floor? We have one question via the app. Can this be beamed up? Okay, we'll leave the question then for the panel discussion. If there are no other questions from the floor, then we'll move to our next speaker. Thank you very much, Dr. Medlock. It's my pleasure to introduce Dr. Peter Gerner-Schmidt. Peter is a Danish-American medical doctor with a specialty in clinical microbiology. He's the chief of the enteric diseases laboratory branch at CDC Atlanta. And he is leading the effort to implement whole genome sequencing for public health surveillance of bacterial foodborne infections at CDC at the United States. But Peter is also involved in its implementation globally through the Global Microbial Identifier Initiative and PulseNet International. Peter will introduce us his presentation that's on next generation sequencing as a tool in foodborne diseases, surveillance, and outbreak investigation, challenges and opportunities. Thank you, Peter. Floor is yours.
Thank you, Marta. First of all, I would like to thank you organizers for inviting me to Parma. It's always wonderful to come back here and get some good ham and some good Parmesan and some good friendship. Now to something completely different. I'm going to talk about foodborne bacterial infections and then the implementation of a new technology, next generation sequencing. And just to start off, because next generation sequencing and a related concept, whole genome sequencing, is often used interchangeable, and I just want to explain the difference, and then I'll just use one of the terms anyway. Next generation sequencing is the technology whereby we are sequencing large amount of nucleic acid sequences, whereas whole genome sequencing is a process that uses the output of the next generation sequences, but also do some post-processing and then also the analysis. So most of what we are doing is actually whole genome sequencing. We just don't stop with the next generation sequencing. So what is the hype about whole genome sequencing? The thing about whole genome sequencing, we are seeing, and this is I believe actually true, we are seeing a paradigm shift. Whole genome sequencing is a truly transforming technology and process whereby microbiology will never be the same again. This is as big as when the Petri dish was invented. And the reason for this is that by using this technology, we can replace most of the methods that we are using in the microbiological laboratory. And think about it. If we had this technology in 2011, come on, there it is. When the Germans and the French and a lot of other countries had this outbreak of Zika toxin producing E. coli of serotype 0104H4, back then it took us about a week before we figured out really what this kind of organism, what it was. It was curious. We were curious. We knew very quickly that it was a strain that produced a bad Zika toxin, the Zika toxin subtype 2A, which is associated with a lot of kidney failure and hemolytic uremic syndrome. But it didn't have any of the known Zika toxin producing E. coli adhesins that you need in order for it to colonize. And it wasn't until somebody figured out to do some PCRs for interogative virulence factors that we identified that. If we had had the sequencing, the whole genome sequencing back then, we would within 2 days have all this information. We would know that it was an E. coli, we would know the serotype, we would know that it was a hybrid pathotype Zika toxin producing E. coli and an interogative E. coli. We would have the full virulence profile. We would know the sequence type of the strain. That's a kind of sequence type that we use for rough grouping of the strains. And we would know the very specific hydrodiscriminatory subtype also. And we would know all the resistance genes that were present in it. We would also know the plasmids and you name it, everything that you can get from a sequence, we would have had it. But I'm going to talk mostly about the utility of whole genome sequencing in outbreak investigations. And this is a slide I borrowed from Celine Nadon from the Public Health Agency of Canada. In Canada, they turned on sequencing for routine surveillance of salmonellosis in 2017. And what they saw was that the number of clusters or outbreaks of Salmonella enteritidis, one serotype, rose by 5 times or something like that. And with enteritidis, it's an extreme example because that's serotype is very clonal. So basically, by all other subtyping methods, they are almost all the strains look identical. But nevertheless, and I want to just emphasize also here, if there are any laymen in the audience, this does not mean that food safety in Canada has become worse in 2017. This is just a surveillance artifact. They are better at detecting the problems. And actually, with whole genome sequencing, and this is data from us, we can also detect the source of the problem more efficiently. We have been doing listeria surveillance since 2013, and with the first 3 years of surveillance, we were able to determine the source of the outbreaks in 3 times more outbreaks than previously. And the number of cases per outbreaks also went down, meaning that we can detect the outbreaks earlier and intervene earlier. And this is what public health is all about. Detect the problems and act. One other thing that we also saw was that if you also monitor the food supply, so this is basically also a One Health surveillance, you will also find things that are interesting and that you will be able to link cases that you wouldn't have been able, or isolates that you wouldn't have been able to link with the previous methods. And this is just the first outbreak of listeria that we worked on in 2013. It's a cheese-related outbreak. It has a French name, but it's manufactured in Wisconsin. All the isolates on the tree that are red, they are clinical cases from 2013. All the green ones are product isolates, and then we have some blue ones that have the same PFD pattern as the other ones. And you can see there are 2 of the blue ones. That is the historical isolates. So these 2 blue that are in the cluster of this cluster, which is the outbreak, defines the outbreak, is actually clinical cases that happened a couple of years before this outbreak. This gave us an indication that maybe this outbreak would have, or has been going on for a couple of years without us knowing. So in general, how sequencing is influencing our outbreak investigations, it improves the case definitions for our outbreaks. It can merge outbreaks by different PFD patterns, and it can also split outbreaks that are not outbreaks that have a common PFD pattern. It increases the confidence in the link between human and product isolates, and it can also, as I mentioned, link historical cases to a current outbreak investigations. And we use it also to characterize the ecology of long-term pathogen reservoirs in the food chain. And that is important also for food safety because you can use this technology for food attribution. Okay, now I explained why it is so good, but as they say, no tree grows into the heavens, and there are also problems with sequencing. And I'm going to explain some basics of sequencing first so you understand what problems really are. The way that next generation sequencing works is basically you sequence the genome of a bacterium multiple times in small random pieces. And then after that, you will assemble the sequences. And when we are talking about the coverage, it basically means what the average number of times a particular sequence has been sequenced in the genome. And usually it's between 20 and 100. After we have sequenced it, we will need to assemble it, and there are two different ways that we assemble it. Either we use a reference reference sequence from a genome that has been assembled, which is well known. It's just like solving a puzzle where you know the image of what it is that you are assembling, or you assemble it without knowing what it is that is actually you are going to look at. And usually we cannot assemble it into one piece, but we can do it every now and then. But most of the time, up to about 200 fragments or contigs. The problem here is that there are many different softwares out there that can assemble, and none of them are perfect, and each of them make some different errors. So you can't just compensate or correct them if you know exactly what it is, the errors they are doing. And then after we've assembled them, we're also doing some analysis. And there are also 2 main ways to do this. There's either you look at it at the nucleotide level, that is the single nucleotide polymorphisms. That is like you're assessing all the letters in a book and comparing the books to each other. That kind of analysis is difficult to standardize between laboratories, or you can look at the gene level. That is what we call multilogo sequence typing, and that is like assessing the letters in the book. So it's not quite as discriminatory as the other one, but considering if you look at all the genes that are present in a species, like all the core genes, you have more than enough. You have several thousand genes usually to look at, so you get a lot of discrimination. But also with this, almost every laboratory have their own pipeline or scheme to do this analysis, and no two pipelines provide exactly the same results. So that means that results generated by different pipelines, they cannot be directly compared. But common to all of them, actually, almost all of them, they generate very reproducible results. That means that in a single laboratory, you can really produce very good subtyping results. But in order to get there, you need to have quality control in place, both at the sequence level, but also at the post-processing level. And as I said, you can easily get some good reproducible results in a single laboratory, or if you have national surveillance where all the results are analyzed, centralized in one laboratory. But if you want to do it between laboratories, there are no international quality standards that exist for this, and there are no international consensus on the use of specific pipelines. And I've mentioned a couple or 4 bodies there at the bottom that is working towards getting to a common goal there. We have PulseNet International that I'm deeply involved in, GMI that I'm also involved in. We have worked with InfoSan, and then I also mentioned the EFSA-funded Engage project that is also working on this. And it has been mentioned, also here, that global surveillance of foodborne infections is extremely important because, as you know, foodborne infections don't respect any borders. You can get, or the outbreak of a foodborne infection can be in a different continent than where you live, and these outbreaks are actually pretty common. How can we solve these problems? Well, you can either go to one of these places in public domain and use the tools that are available there. And I've mentioned 3 examples here. We have the Center for Genomic Epidemiology from the Danish Technical University that houses a lot of different tools that you can go and use. Another EFSA-funded project, the Innuendo project, they've also developed some software that's available to the scientific community. They are also very good. And then the NCBI also have developed their own pathogen detection pipeline, which also is a very elaborate system where you can basically analyze all your data once you have uploaded your raw sequences to their sequence read archive. The problem there is that you need to upload the raw sequences, submit them to the public archive. So you need to put them in public domain, and that will hold back quite a lot of laboratories from using this tool. And the problem with the other 2 is that the servers are probably not big enough to handle the volume that is necessary. Alone in the United States right now, we have this year sequenced about 50,000 genomes and submitted that to NCBI. If we need to analyze all that, most servers will go down. You can't do that. But NCBI can do it. So in order to do whole genome sequencing at the global level, there are a few things you need to consider, and this is work we've been doing in PulseNet International. We need to have some tools that are fast and precise and are easy to communicate, and where you also can share the data. If you can't do that, you can't do international outbreak investigations. You don't have to share all data in public domain, but at least the tools you use, they need to be standardized or harmonized and validated against each other if they are not standardized and identical. So you know what it is you're doing and how the different methods compare to each other. The results will need to be generated in a standardized format so anybody anywhere can interpret the data for you. The volume needs to be low volume because in developing countries they don't have high internet speeds and you need to be able to communicate all over the world. And ideally you don't want to be forced to go back to the raw data because that is where the heavy computational power is needed to do the analysis. And what we are looking at is solutions that are practical but works. They don't have to be perfect. If it works, it's good enough. And this is just a slide that shows the PulseNet model that we are working on. We are trying to put all the computational facilities, all the hard work, that needs to get out in public domain so it's available to the whole scientific community. And ideally NCBI or EBI or one of the other big ones that really can handle everything. But then the data submitter will have to have, at least for now, the option of sharing also the raw data with the public, or at least keep it in a food safety community database that will be accessible to others that could have interest in looking at it. So basically all the members of PulseNet International, for example. Go on. Then the next problem we are facing is the challenge of the data interpretation. With whole genome sequencing, defining how close is close, that's not an easy task because no isolates are 100% identical and the data are contiguous. So this tells us, or it should tell us, that epidemiological data and other metadata are more critical than ever for whole genome sequencing data interpretation. And I'll give you a couple of examples of that. This slide shows some results from an outbreak investigation we did in the United States this past year. We had more than 200 cases of patients that were infected with a salmonella, or not with one salmonella strain, but with strains of 6 salmonella serotypes. And it was associated with the use of kratom, which is an herbal supplement. It's an opioid agonist, meaning that it's something that drug addicts can use to treat their withdrawal symptoms. And it's not regulated in any way, and we're discussing if it should be regulated. Maybe it should. We detected it as a monophasic Java strain, 4512B-, and as you can see, yep, it's pretty clonal. 0 allele differences by core genome MLST. So it's very simple. This is definitely an outbreak that's going on, and the green ones are actually isolates from the implicated products. This is actually also a product that is impossible to trace back to more than just the region. This product is imported from Asia, but nobody can tell you where it came from in Asia. It's impossible. And that might also explain a little bit why it's so diverse with the salmonellas that we are having here. In contrast to this, one of the other serotypes, I'm only going to mention one, and that is the Okedi outbreak, or the Okedi part of the outbreak. Look at that tree. Does that look like an outbreak? No. The whole genome there is no clonality in this. You have 4 clusters that varies up to 24 or 28 alleles within each cluster, and between the clusters up to 77 allele differences. That is not an outbreak. Unless you look at the serotype, Okedi. Who has ever heard about Okedi? We had not heard about Okedi, so we knew this would be an outbreak, but we hadn't considered this at all until we saw that FDA had isolated this serotype from this product. and you can see the product isolates, they come in all the different clusters. And lo and behold, of course, all these patients had also been consuming kratom. So this is an example of some of the limitations of whole genome sequencing. If you're only using whole genome sequencing, we would not have detected this outbreak. There was also more than one PhD pattern involved. So in this situation, the serotype would be helpful because it was such a rare serotype. But otherwise, it would be the traceback information from the other part of the outbreak, the Java part of the outbreak, and the interviews that were helpful here. And yet one more example, you should never let the sequencing data fool you. You have to consider all the supporting information. This is a different outbreak that happened around Christmas time last year, or rather it's probably going on still, because this is associated with a reference strain of Salmonella typhimurium, an ATCC strain that is used by teaching laboratories and also by some reference laboratories every now and then. And these are students that have been infected, and then suddenly FDA isolates a strain that is indistinguishable from eggnog. It's around Christmas and people are drinking eggnog. None of these patients had been drinking eggnog of the implicated brand at all. So this is clearly a spurious finding. So just because you have a match between the food isolate and the clinical isolate doesn't mean a thing. Doesn't have to mean a thing. Okay, then we have a challenge of data sharing. Of course, it's important that we share the data in international outbreak investigations. If we don't share the data, we will not be able to investigate the outbreak, figure out how big they are, where the problems are, and you know that the example down there at the bottom is a listeria outbreak that was published this year involving the frozen corn product. And there are some salmonella outbreaks that have been, or listeria, the big one in South Africa, and a salmonella outbreak that has been worked on by Infosan. So we need to be able to share the data, and ideally they should be shared in public domain in real time at the SRA, ENA, with some minimum epidemiological data, time, place, and type of isolates. But there are some problems for many countries to do that, and some of the barriers are ethics. You can't share personal identifier, and of course then there are some problems with intellectual property rights and other legal issues. And then when you're talking about food safety, you would also like to see some data from the food industry, but they are also a little bit hesitant sharing the data because there are no statute of there's no statute of limitations on liability. So if data can easily be abused, and there's no precise definition of an outbreak. What we are seeing right now, some of the outbreaks we are seeing have very few cases every year and can go on for years. So 2-3 cases dripping all over the year, and then the same number next year, and then again. And then sometimes there might be a product isolate also that matches something that happened 3 years ago or 2 years ago. Then the industry says, well, what then? Should we really do something? And we need to figure out how to deal with that. There is no international interpretation standards, and that means there is also room for misinterpretation of data. And if there is misinterpretation of data, that could also be used for creating trade barriers. There are a few things that remains we need to think about, and this is my next to last slide. WGS turnaround times is still an issue. In routine, it takes about 7 workdays for us to work through the whole process. There's still the cost of sequencing. If you're only doing the sequencing, you can probably do it for $50 or something like that. But if you also have to consider the DNA preparation, the library preparation, all that, you're probably close to speaking about $150 to $200 per strain. and that is pretty expensive. But if, of course, you consider that you're not doing just subtyping, but a lot of other things, well, then it might be cost efficient. Then there is the problem of, since we are detecting so many clusters, which ones should we investigate, because we don't have resources to do them all. And then finally, we are seeing more and more clinical laboratories are detecting the diseases by using PCR methods, so we don't get any cultures, so there's no isolates we can sequence. And that leads me to the final slide here, the bright new reality that we are going to see in a few years' time, where we are not going to sequence the isolates, but we are going to sequence the specimen, including the genome from the patient by metagenomics. So we're sequencing everything. And with that data, we can also link that to data from other sources, including non-lab data. If you have a register that you can link to a patient, well, you can do that. And then you can really get some big data and a lot of information you can use for a lot of things. But of course, because you are also sequencing the patient, you have privacy issue, you have regulatory hurdles you need to pass, and you have data capacity because the amount of data you're looking at is enormous. Especially the two first is something we need to begin to look at now. Metagenomics is not mature as a technology yet, but we need to have solved the privacy issues and the regulatory hurdles before we do this. And with this, I think my time has run out, and I am done. Thank you.
Thank you, Peter, for this very interesting presentation, identifying also the pros and cons and the challenges that we will need to face in the coming future. Are there any questions? Peter? Yes, Alessandra.
Regarding this rare serotype that you saw so many differences in alleles, can you believe that there are some species, we learned this from pulse field, there are some species that have more conserved genomes between the same serotype or serovar, and others that are more variable even in a short period of time. So we should trace back exactly what we learned from pulse field variability probably into genomics interpretation. So I remember when you consider pulse field gel electrophoresis from E. coli and when you consider from Salmonella enteritidis. So
oh, yes.
we must have this reference in our mind also valid for whole genome sequencing interpretations.
Oh yes, that is why we can't come up with some clear-cut cutoffs for what defines an outbreak. It depends on the clonality of the organism, and as you say, even though we can differentiate within Salmonella enteritidis now, the levels of differentiation here with enteritidis, if you have 7 allele differences or 7 SNP differences, if you see that in Listeria, you say, oh, this is an outbreak. But with enteritidis, it's definitely not. There you're down to 0 to 3 or something like that. So there are, you need to know how the organism looks when you are interpreting your data.
Another question here from the floor.
Thank you. Thank you, Peter, for a beautiful talk and explaining so clearly how this is a revolution in microbiology. You made a very strong statement, that is that whole genome sequencing works with a centralized analysis. I remember in the good old days of PFE, if I'm not wrong, CDC decided to decentralize the analysis of PFE because it was overwhelming to do it all centrally. Now with these new technologies, we are seeing that it's very difficult to have consensus pipelines for analysis, etc., etc. So with your knowledge of Europe and your knowledge of the USA, could you give us your view on how feasible you see this central analysis for the whole of the European Union, for example? Thank you.
I don't think you can do centralized analysis with sequencing, and we are not doing centralized analysis of PFGE. That is also decentralized. But in order to do it, you need to either standardize it or you need to harmonize the methods. And if you're only harmonizing, you need to know exactly how the different methods that you are allowing in your network, how they compare to each other. And you need to take that into account when you develop your interpretive criteria. And it's everything that I've been talking about here that you need to validate. And if there are any of these variables that you are looking at, for example, if there are some of the assemblers you know is not going to work with any other assemblers, then you just don't use those. You will say, well, we are going to use Spates and we're going to use Velvet and that's it. Or Spates version 2 and Velvet, and then that's it. And then the same with if you're doing SNP analysis, you have to validate the SNP pipelines against each other. And even, I would say, with SNP analysis, if you really want to draw a common tree, you would need to use one pipeline to draw that tree. With whole genome MLST, it doesn't matter that much as long as the allele calling you use has been validated good enough, as long as you have a common allele database.
Also, my co-chair has a question.
Yes, so I just abuse the fact that I'm at the stage here. There are a lot of close matching on whole genome sequencing is creating a high degree of confidence that a source has been found. So I was intrigued by your eggnog example. And do you think that was a genuine case of coincidental parallel evolution to a similar or identical type, or was that a pipeline issue?
I think there must be a question of somebody who contaminated the eggnog at the plant or something like that. Somebody who has been infected, but I don't believe in parallel evolution that is converging there. I don't think that happens.
Okay, thank you.
We have also a question from the application that it's to Peter. What do you believe are the main challenges in validating whole genome sequence methodology?
Well, the main challenge right now is actually to do it and also figure out what it is that you want to validate. And that is what we are doing in PostNet International now. We have created some working groups that are looking at both the assemblers and the different allele schemes that are around there to figure out how we can do it most efficiently. It would be very nice if we could reach consensus about everything and standardize everything, but we can't. There are too many people who has their own pipelines now, and they're not giving up.
Thank you very much, Peter. And for your presentation. And now I pass the microphone to Mike for the moderation session.
Thank you very much. So first of all, can I ask all of the presenters to come back up on stage, and also 3 colleagues from EFSA, who I'll introduce in a second, please. So you've been introduced to the presenters who have all given excellent talks, and David, there is a chair on the end here. Yes, it's. But can I just introduce also, we have on the panel, we have Dr. Ernesto Nibani, who is the acting head of EFSA's Biological Hazards and Contamination Unit. We have Dr. Nicholas Chris, who is the head of EFSA's Animal and Plant Health Unit. And we have Dr. Tobin Robinson, who is the head of EFSA's Scientific Committee and Emerging Risk Unit. So an esteemed panel we have in front of us. Now, my role as moderator, I've been told, is to somehow briefly summarize the key points that have come up across the presentation. So I will attempt to do that in as short a time as possible, because I think it's really important that we get a chance to get the views of the panel, interrogate the panel. I'm not quite sure which you want to view it as, but I found it a fascinating session. So we first of all heard about how globalization brings complexity. It also introduces particular drivers of foodborne illness, I think it's fair to say was part of the thesis, but the control of those drivers may be problematic. And I think there are a number of questions that come from that. For me, is it the role of the risk assessor to consider the problems that David highlighted, or is that a downstream issue? Does it still make sense to undertake risk assessments in this globalized age at a national or even a continental level? Then we heard about new genomic typing techniques, providing new insights into antimicrobial resistance and its mechanisms of spread. I think the question is, how can we use this to inform control and prevention strategies? Do we yet really understand the importance, the relative importance of animal and foods as a determinant of human antimicrobial resistance? And perhaps an even bigger question, which came up with the third presentation, is the role of the environment in antimicrobial resistance. I see antimicrobial resistance as the biggest threat to human health posed in the infectious disease environment, and I think the environment is the big question. We also heard this afternoon about the potential for plants to acquire antimicrobial resistant agents or components from the environment. So some of the questions that came to my mind is, why are some plasmids epidemic and others not? Is that to do with the plasmids? Is it to do with the organisms? Is it to do with the vehicles that transmit them? What level of threat does uptake by plants represent to humans? And what are the relative importance of human-animal sewage in actually what we see in the environment in terms of antimicrobial resistance? So how much of that is because it's come from animals and humans through the sewage system, and how much it is because there are residues of antimicrobial agents which are providing selective pressure within the environment? Vector-borne diseases. Julien gave us a tour of vector-borne diseases in Europe, and I think for me the take-home message, it's for many parts of Europe that may not be seeing at the moment, the question is when, not if, they will actually see the emergence, or there'll be an increasing number of vector-borne diseases, and climate change may accelerate that. So how do we monitor or assess the risks? Do we need more data? Do we need different data? Can we make use of big data? And given the ever-changing picture we've seen with vector-borne diseases, how can we balance the need for rigorous scientific processes for our risk assessment processes against the ever-changing environment and the fact that we need information now to drive policy decisions now? And then finally, the last presentation, opportunities and challenges of whole genome sequencing, next generation sequencing. Take-home messages for me, the need for standards. There are a range of problems, data sharing problems. Cluster triage, this is a real issue, I have to say, for us in ECDC, is that we are now facing the prospect, if we see the same experiences in Canada, of actually seeing the number of clusters detected completely outstripping our epidemiological capacity. Now, that raises ethical, legal, and other issues, so I think that's a real challenge. But of course, there are many benefits, huge benefits that come from it. So that's just a quick whistle-stop tour from my perspective of some of the issues. We do have, can we put up the questions that came through? There are one or two that we haven't yet addressed. And then we're hoping there'll be questions from the floor. So while we're waiting for the questions that came through on the app, can I ask if there are any questions from the floor? Please. Microphone's just coming.
Yes, right now I still have one, and this is regarding also the power of whole genome sequencing data. One of the key issues that we have is the source attribution also for antimicrobial resistance. and in which concerns pathogens that cause acute diseases, this could be more easy. For instance, if we are talking with E. coli resistant to antimicrobial resistances, I think that right now we don't know how many SNPs could occur during the colonization of our gut. And this seems also to occur to other bacteria that we use to trace the origin of the antimicrobial resistance infectious agent. So I'd like to have really, from your point of view, if really this is right now a challenge, is if this for E. coli, Staphylococcus aureus, we know right now that are maximum, let's say, of SNPs that we should consider in order to correlate as the same, to identify as the same strain. I don't know if it was clear, but mostly my concerns is regarding not the pathogens that we are able to immediately identify that are causing a disease. The transmission was occurred in a short period of time, but ones that can, for emergency or for collection, could have been colonizing, adapting to different settings, as instances, the gut of animals, humans, or even the environment. And how whole genome sequences could address this topic. Do you have possibly number of SNPs to consider, a minimum? How can you solve this issue?
So I think that's probably for Alessandro in the first instance, the question. So it's what thresholds should we be setting if we want to use whole genome sequence for source attribution for antimicrobial resistance? Can we be setting thresholds for source attribution purposes?
Of course, if you imagine that the horizontal transfer complicates the analysis, because you cannot attribute antimicrobial resistance origin to a species if they move the plasmid from a species to another. However, I think it's you can attribute if you are sequencing single clones, as is showed, and this is, of course, important if you can demonstrate that the plasmid is alive, it's making conjugation. So sometimes we do extra work beside the sequences to demonstrate the plasmid is conjugative, highly conjugative, that is broad strange, that what you observed can be confirmed by experimental evidence. Then you have a series of information regarding your question, why some plasmids are epidemic and others are not. The first is these plasmids confer an important and relevant resistance. So this could be the first characteristic that has promoted their spread. But not only, because you see sometimes the same resistance gene on two different plasmid types. One is more diffused than the other. So again, from sequencing analysis, you can see that there are other characteristics that can be found on the plasmid itself, like stability, persistence, host range, frequency of conjugation. All these characteristics are conferred by the plasmid to the resistant gene. So it's possible that the resistant gene has more success because it was associated with a very efficient plasmid.
Thank you. Yes. Peter, did you want to comment at all on the issue of antimicrobial resistance and the role of sequencing in improving our understanding of the dynamics, the spread of antimicrobial resistance?
I think Alessandro has covered it very, very well.
Okay, good. I don't know if any of the colleagues from EFSA want to comment on that particular issue. Ernesto.
Just a very quick question, since I have Alessandro next to me, and I think I've always been intrigued by this epidemic of plasmids. Have you ever thought about any genetic strategy to really fight the spread of the plasmid? Because if you had to control it in a hospital or in any environment, not targeting a given bacteria, but targeting the plasmid itself, how would you do it?
Future frontiers, okay? What I say now we can be really contradict tomorrow. There are several approaches that we call anti-plasmid. It could be a medicine of the future. The idea is to cure the plasmid from the bacteria, to avoid to kill the bacteria. This would be really an ecological approach. But how to reach these molecules without perturbing the microbiota or whatever is a challenge. And more we know and more we can be close to this kind of medicine, which for me, it's, of course, it's a Nobel Prize, this one. I mean, if one is able to cure a plasmid from a bacterium, it's an exceptional goal, okay? And I know there are several molecules that have been screened. There was a European project financed by EU in which I was the reviewer, so I was not participant, but there was a work package on that. So to screen molecules preventing conjugation. It's the pills, the anticonceptional pill for bacteria. And I think they had some molecules on the pipeline. So just to give you the idea that this is really the frontiers of science, but there are very few groups working on that, so we'll see in the future, okay?
Thank you, that was a great question. So we have some questions on the board, and I suggest that we run these through. So first of all, Alistair Witherspoon's asked, the platforms for whole genome sequencing are evolving, constantly changing new versions. So we have, for example, the Minion technology. What challenges does this present to acceptance of whole genome sequencing into routine use?
I think the only challenge that introduction of a new technology faces is that we need to validate it against the other ones, so we are absolutely sure that we can compare the data, the current data, with what we had in the past. I actually see it more as an opportunity for the market right now is almost 100% Illumina, at least for surveillance. And that means they also control how much sequencing costs. So the more competition we can get, the better. And hopefully that will drive the cost of sequencing down. And MinION, which was mentioned as a specific product, is something we are looking very much forward to because it's the longest of the long-read technologies. And we need some of the long-read technologies when we are going to move towards metagenomics more than ever.
Yeah, I mean, I think the point about competition is important because ultimately economics, as in everything else, will drive this to a huge extent. And I think that actually, if it drives down costs, it's likely to increase the take-up rather than inhibit it, one would hope. So let's move on to vector-borne disease. As ad hoc raising awareness is key to preventing these diseases, if this temporarily triggered by predictive surveillance, could the current outbreaks in southern Europe be controlled? And I'm not sure whether this is from Alessandro Broglia, but I think for me that brings into the idea is actually can we be looking at projected changes in climate, land use, environment, temperature to predict how the vectors will spread, how people will be exposed, as well as drawing trend lines, I guess.
Yeah, I think with all vector-borne diseases, I mean, if we don't engage with the public themselves and give them the information they need to take necessary precautions, then we're onto a losing one already. I started my career doing malaria control in southern Africa, commissioned to do impregnated treated net programs, and quickly did a survey of the local population, found that less than 30% knew that malaria was transmitted by mosquitoes. So if you start distributing an intervention where 70% of the population have no context into why they need to retain those bed nets in the face of a natural disaster, then you're not going to win the game. And actually, after 6 months, we had 95% retention. So I think engaging with the public on all matters in relation to disease control is crucial, particularly for the Aedes mosquitoes, because actually it's people harboring the mosquitoes around their home. And if you haven't got community engagement, then you can spend all the money you like on insecticide treating or using GM mosquitoes. But if you haven't got individual and community engagement, then it's difficult. West Nile virus is slightly different in the fact that humans are dead-end hosts, so they're not a source of infection back to the mosquito. You can collect huge numbers of mosquitoes and not find much evidence of the virus in them. And then, of course, you've got birds potentially as sentinels. I think, yes, sharing information, particularly not only to the public, but also to local authorities in a timely fashion, can target vector control and also targets of repellents and avoidance of mosquito biting.
Thank you. I don't know whether Tobin or anybody from would like to comment on that. David.
It wasn't in my talk today, but I've been involved with Nick Ogden and other people looking at the spread of Lyme disease and the spread of West Nile virus in Canada, and with the people in the lab in Winnipeg. And certainly there's a lot of back and forth with the public on landscape changes, on actually design of neighborhoods and which kind of neighborhoods actually promote. If you have wide roads with storm sewers underneath, the mosquitoes overwinter, those kinds of things. So there are structural things that you work with, and it also raises the profile among physicians. So for many years, there were adventitious ticks dropping off, and there were probably cases of Lyme disease, but they were not diagnosed, and the people who got it were dismissed as being paranoid, and some of them probably actually had Lyme disease. So as that awareness goes out, people are testing, the tests are getting better. So there's a lot of discussion with the public, with the medical community, with the public health people.
Yeah, just to add to that, with Lyme, I mean, when you've got a situation that's changing over time, the continuing education for clinicians to ensure that they recognize that Lyme is now an issue is really important. I also want to mention behavioral science as well, because as scientists, we tend to write things in very black and white scientific text. This is the case. Actually, that can really turn people off, or it can scare people. And one of the things we're trying to do in the Caribbean, actually, is to try and get information about mosquitoes in the national curriculum, so the next generation are educated about future threats.
Yeah, I mean, I think that, it's very much in keeping with the theme of the overall conference. And I think that there are some nice examples of citizen science projects, if I can call it that, citizen science projects which have been looking at vector-borne diseases. And I know that, for example, in Spain, they're very actively engaged with the hunting community to send in ticks, easier with ticks than mosquitoes. Again, any other comments from colleagues? Yes, Tony.
Yeah, maybe two on this. Firstly, coming back to the desire that we have, I think, to develop rather more predictive approaches to some of these diseases rather than simply responsive ones. We have a problem, how do we manage it? So some interesting work really in the area of foresight technologies, and particularly in the area of vector. There's one example which I think is rather nice of the type of thinking that we may need to develop, which is going a little bit away from our comfort zone and how we address things. This was an exercise which, if I remember rightly, was carried out by DEFRA in the UK a couple of years ago, where they did a bit of brainstorming about some future scenarios of what could happen, particularly in the area, in this case, of vector-borne diseases. And it's coming back to the concept of drivers. So what are the things that are changing the environment to which we're being exposed to? One of the drivers they were considering was the economic crash at that time. So how could the economic crash affect food safety, vector-borne diseases, these sorts of things? And after a bit of mad thinking, one of the scenarios they came up with was, due to the economic crash, people would be abandoning, for one reason or another, second homes, holiday homes, and homes in southern parts of Europe, where often these are associated with swimming pools. So as a consequence, you have an increase in the number of swimming pools which aren't being properly maintained. And one of the potential consequences of that is an increase of breeding of things like mosquitoes. And a very nice scenario, they actually went to look to see whether they could find some data to back this up, and yes, there seemed to be some indication that this was indeed quite a viable scenario. So the example, I think, is nice, but also I think it's a good illustration of the fact that we need to be thinking a lot outside the very simple kind of areas that we look at, so how food is produced, for example, and very simple ones about the environment and how that affects vectors. I think if we want to anticipate the challenges that we'll be having in the future, we need to go wider into more socio-economic factors that have an influence on our safety.
Okay, thank you. So if we come back up to the questions, I mean, I think just keeping to the theme of vector-borne disease for the moment, Sophie Darnders asked, and I think some of this has been addressed, but not all of it, how can vector control be integrated in environmental risk assessment, for example, considering changing habitats, climate, increasing resistance to products? How can we increase preparedness for new incursions of vector-borne diseases, considering globalization, changing social, ecological, economic realities, and keep this high on the agenda? That's a small question. Who wants to go first? Jolyon.
I'll have a go because I've been thinking about this for a while. But I think in the UK we have a climate change risk assessment, and within that there are various themes. One is human health, another one is biodiversity, another one is infrastructure. And actually, if you move from one room to another, you can listen to future human health risks from infectious disease. And in the next room, we can be talking about, and also the benefit of urban green space for health and well-being, access to the countryside, reduction in mental health. And then the next room, it can be about making sure that we have bridges between fragmented habitats. So when climate changes, insects or birds or plants are able to move along wild corridors, green corridors or blue space. And there's very little sort of consideration of actually whether there is an impact either way. Everyone thinks about the benefits of biodiversity perhaps to human health, but are there any sort of maladaptation issues that we need to consider? So by creating better urban corridors, are we allowing wildlife to move into urban areas? Are we allowing deer to move into urban areas? I mean, there's another issue with Echinococcus multilocularis and urban foxes, for example. So if you've got deer in urban areas, then do you suddenly present an issue with tick and Lyme risk? And what you want to do is develop a strategy that has considered all these before you even embark on urban green space management. So actually then you can be thinking about how you can simply manage vegetation. In urban areas, people go around and cut the vegetation at certain times of year. If they cut it at a certain time in southern England at the start of April, it would minimize human exposure to Ixodes ricinus ticks and therefore minimize the likelihood of sort of Lyme transmission. On these sort of large-scale programs for habitat creation, I mean, I do think that within the environmental impact assessment, when you're considering the health chapter and what risks there might be, often maybe mosquitoes aren't an issue, but if they're considered in the early stages, then it will preempt any questions to avoid, as I mentioned in my talk, people sort of opposing them. But one thing I think is quite key is that if you talk to people at a local level about preparing for mosquito and tick-borne disease, they'll say, we have enough priorities already, and we don't have the money to spend on this new priority. So actually, how can you get them to engage in that? What you can do, and in the UK we have a national risk assessment. So we get vector-borne diseases on a national risk assessment and saying this has been considered and you need to have a local contingency plan for it, then suddenly there is a priority on the list that comes from a different angle rather than from public health.
Okay, thank you. Now I see Nick has grabbed the microphone too. Brave man.
Thank you very much. Now first, I want to ask a question. How do these flies know who's got the microphone? Because they do.
Indeed.
Must be that. And thank you, Peter, by the way, for not ruining eggnog for me, because winter season is coming. But regarding the question from Sophie, and especially also how do you keep it high on the agenda of risk managers, and how do you keep interest in it going? And of course, from EFSA's point of view, together with the ECDC, there was VectorNet, but I'm in the fortunate position of being head of unit animal and plant health, and in plant health, vectors are also important. Xylella fastidiosa springs to mind, and we also have entomologists. But it goes to show that in the time of glyphosate or African swine fever, incidentally also vector involvement, it's very hard to keep interest in financing, exactly what Joylan said, financing collection of data attractive and keep the money flowing into it. So again, Joylan mentioned the word of collaboration, because there is data out there, there's research out there. How do we share it population data, how do we share it? We also work with the JRC and the European Satellite Center to get more on the environmental aspects and try and keep that high on the agenda. But again, the one thing, and this will come, I think, in the session tomorrow, of evidence management. How do we manage and continue to manage the evidence and data that we have in vectors as well? Of course, it's not so difficult now to sort of keep the interest of risk managers, but in terms of getting the money for it, you're absolutely right. We find that also very difficult because the money tends to go to the momentary crisis. And that's exactly where Tobin came in and said, but we try and reserve a certain amount of our budget, of our funding, to keep going in areas of crisis preparedness. But it's a tough call.
Sure, absolutely. I don't want to let go of the question that I asked one of the summary is, do we have a clear view? Does EFSA have a clear view of the respective roles of animals and the environment in terms of antimicrobial resistance? This is, I think, another small question, but I think Ernesto has obviously got the microphone.
You just gave me the perfect opportunity to lobby for my favorite topic now. Of course, we don't. Yeah. We don't, because we don't have barely any data on antimicrobial resistance in the environment. So we are at the moment of reviewing, mandated by the European Commission, the way we do harmonized monitoring. And the question is, do we spread the commodities or animals or reservoirs we survey or not? And this is also put in the international context, a very delicate topic. Also, Codex International is reviewing the topic, and we seem to have half of the world wanting to find out what is going on in the environment, while you have the other half of the world, including a very powerful player there, like the USA, saying that they just want to follow a risk-based approach. Basically, unless you can prove that the environment is a risk, they are not ready to start monitoring. But how can you prove that something is a risk if you lack the data to conduct your risk assessment? So it's a vicious circle. So I think we have been seeing today in different talks how important it is to find out what's going on in the use of water in agriculture, the food of non-animal origin, aquaculture, etc., etc. And we basically have no idea of what's cooking there. Don't know if, Alessandra, you want to say anything else?
Do you have the answer?
No, I don't think that we have data enough for the moment to evaluate this, and we need to do some program of surveillance, no? Just to collect data very objectively.
Okay. Now, I want to also... Aha, please, yes, we have another question in the front row here.
Yes, hello, Beatriz Guerra, also from Biocontact Unit, from Ernesto Unit. Working with whole genome sequencing data, I suppose ECDC and EFSA CDC, we face that to extract data on antimicrobial resistance, we need to have a curated database with genes. If we want to find virulence genes, we need curated databases. And we have discussed very often, okay, if you don't curate the databases, the data that we extract using different tools are different. I think you, Alessandra, are curator of the PMLST database. Which are the challenges that you find there? And you, Peter, you are user of this database. What are your problems with these databases? I don't know, to say some examples, ResFinder, CART databases, and your PMLST or plasmid databases. Do we need central curation?
Yes, please. No, I mean, you invent something and you don't know that your life is signed forever, because I am curator of PMLST for I1, F, HI2, plasmid types, and also plasmid finder now. And so you see some email in the morning in which you have a list of new hypothetical plasmid types, and you have to check if they are true and to assign them to groups if groups exist. So it's an enormous work. For Plasmid Finder, the Genomic DTU has a centralized group, so they are really helpful. For PMLST, no, but this is true also for all the MLST curators, not only for plasmid, also for MLST. So you have the support of Oxford, but you are responsible for what you put inside as a batch, as a single isolate. Is an enormous work. So sometimes, I'm sorry, we are not daily updating everything, but it's like this. Yes, of course, it would be something like a European project to maintain this database, always updated, available, and also talking each other.
Yeah, I would add it's extremely important that the databases that contain specific genes like virulence genes or resistance genes, they are highly curated. We need to be absolutely certain that what we are detecting is what it is we are detecting. So that is why Alessandra is extremely important. But on the other hand, with the allele databases for the MLST systems, not the 7-gene housekeeping gene MLST, because that is similar to the virulence gene databases, but the other ones there where you have several thousand genes, it's not feasible that you can look at all the variants that you detect. You have to rely on automatic detection. And I don't think it matters much if there is an error every now and then in a single gene, because it will be diluted out when you do the analysis anyway. If you have 6 allele differences or 7 allele differences, doesn't matter.
Okay, so if we could have the questions up, because I would like to come back to, yes, actually I think it was the first question that came in from Matti Aho. And I think this picks up a bit of what David was talking about as well. Solving food safety problems at the source of raw materials and products may be the desired option. However, our policy tends to support import restrictions at the EU border. Seems we are not willing to solve food safety problems at the global level, are we? And I think this touched on where you finished, David. And I was also minded, so what you explained to us, what you described to us was a complex system. And I'm minded that the director of EFSA in his opening talk said that we are facing more complex problems for which we need more complex approaches. So I guess the question for the panel that I would put is what collaborations do we need to see in place to tackle what are now global food safety issues? Ernesto?
Well, I just want to react a bit, because at least in the antimicrobial resistance front, I don't recognize this as a true statement. We don't have any policy instruments to stop anything coming into Europe, simply because there are no such criteria in legislation. And for some time we have been suspecting that is, of course, a point of entry for many things. We take great care in controlling as much as we can the problem, but not the same happens everywhere. And we have basically no clue about what resistance genes or resistant bacteria come in on imported food. So again, in order to be able to know this, we should be first getting some data, and then we can start talking about this. Another thing is that in this particular front, we have seen also many talks yesterday and today talking about the global problem that antimicrobial resistance is. And of course, if we don't spread out the wisdom and try to implement similar systems in other places, we are lost. And this is where we have a role to play, I believe. But I think that the statement is not correct. I wish we could stop a carbapenemase-resistant contaminated piece of meat at the border, but we cannot.
Okay, it's good. Always good to have a provoking question. David?
Even within Canada, if you look at ground meat that comes, and you trace it back, and it comes from a feedlot, and the feedlot has cattle that come from all over Western Canada. It's almost impossible to trace back the origin. And then if you do that internationally, you run into serious problems. I guess one of my frustrations, even when I started working on foodborne and waterborne diseases 30, 40 years ago, was that we have all these scientific studies, we could describe things, and then we as scientists would give the information to a politician or to somebody in public health agency and expect them to integrate all the information. And somehow they're supposed to be, we expect them to be smarter than we are, right? Which is, sometimes it may be true. So then I try to figure out how do we integrate it, and at some point you find that you can describe these problems, as Silvio said in the opening session, with scientific information. You can trace back and see where it came from, but the solution is not per se scientific, right? So you build infrastructure in other countries, you look at how you monitor things, and there are scientific elements to that. I was pleased, Peter, that you clarified that Canada didn't necessarily have more foodborne disease. It was monitoring, and that, of course, that's a caveat on all those graphs that I had, because our surveillance systems changed over that period of time. So were those real increases? Were they not? I think there's so much uncertainty in there, which doesn't mean we shouldn't do anything, but it means that we need to make decisions with the best information we have, and then we can't stop things at the border, which means maybe we need to be working in other countries to build up capacity. I mean, OIE has tried to do that with PVS, and I've been involved in some of those, which can be incredibly frustrating because there isn't money to back up changes in terms of what's going on in other countries. I don't have an easy answer, but we can't solve them just here, and they're not just scientific. The science contributes to it, and it helps our understanding as to what our options might be.
Okay, Peter, did you want to comment?
I'd say, at least for tracing foodborne outbreaks, at the public health and the food regulatory level, we have systems in place where we talk to each other all the time. We have networks where everybody knows each other, laboratorians and epidemiologists and food regulators, and we talk to each other all the time. But that doesn't solve all the problems, and I want to focus on one thing that we have found very useful in some of our outbreak investigations in the United States, and that is talking to the industry, because the industry often have some information about distributions and origins of things that we don't have.
Okay.
And they are willing to share that information. Sometimes. Yeah, maybe sometimes.
Sometimes. Okay. Please, Tobin.
Well, just to give a slightly different perspective on the question, which is pretty much pointing to a fortress Europe. I would say, again, it's coming from the emerging risk perspective, where we have new challenges coming into Europe, coming onto European territories, particularly in the areas related to my colleague here, of animal health and plant health, but not only, where we are seeing diseases that we have very little experience with in Europe. And here, in fact, the expertise is outside of Europe. So we really need to be going to other people to benefit from their knowledge. And I think we ought to see that really as an opportunity. It helps, I think, with the exchange of knowledge, not only what we have on the challenges that we're used to facing, which relates, I think, in some way to the question that was posed on how can we help improve food production processes outside of Europe. But this would give us, if you like, put it on a more even playing field, rather than Europe trying to teach the rest of the world how food should be produced. I think it produces a more collaborative environment in which we can exchange usefully on expertise that is distributed around the whole world.
Thank you. I see 0000 on our clock, so we have come to the end of this session. So before Marta comes and summarizes all of this for us, just like to thank the panel for answering the questions, putting yourselves in the firing line, and braving the flies. And with that, if I could ask you to join me in thanking the panel, and then Marta will summarize for us.
Thank you, Mike. So thank you also for being with us despite we're a bit late. Yeah, I think this session today has addressed what we're really intending to address, and has shown also that the One Health approach is really the solution for the challenges, the threat that we are facing, and also the cooperation among different disciplines, breaking silos, working together, and this transboundary on the areas of knowledge. But we started talking about globalization and foodborne diseases, and also how we got some achievements, but also some unexpected outcomes, how the complex is, but the complexity should not stop us working, and also identifying some challenges for policy as different responses to globalization and foodborne diseases will have different impacts, some more positive than others. So we need also to bear with that. Regarding antimicrobial resistance, a big threat to our health, we have also learned the complexity of the epidemiology, and also how the spreading routes for genes transfer are complex and global. And also the relationship between environment, wastewater, and antimicrobial resistance, how there is a need for some standardized criteria in environmental sampling, how the efforts in understanding the role of environment in antimicrobial resistance are needed, and also, for example, how would be the uptake of veterinary drug residues by different crops, and there is a lack of risk assessment for irrigation of vegetables with wastewaters, also how prioritization of microcontaminants as per the uptake on crops is needed, as well as criteria for controlling irrigation water. And also don't forget, as our speaker reminded us, to the non-viable, non-culturable bacteria. Moving to also vector-borne disease, this was also a clear example of the One Health approach to inform policy, the multidisciplinarity to be successful, and the importance of the active and continuous surveillance, maybe also using crowd surveillance, the crowd citizen, to also help in this network sustainability in case of no resources are available. And also the conflict between biodiversity goals in infectious disease health risk, and also the need to include vector risk assessment in environmental impact assessment. Being proactive instead of being reactive was also something that the panel highlighted, and also the foresight efforts in anticipating these threats. Then we move to whole genome sequencing as one of the new tools that will help us in be more quick and respond better to outbreaks, despite that this might also pose communication challenges, but also that this tool will also give us more evidence base for food attribution on the area. Also, regarding whole genome sequences, different challenges were identified. Also on data interpretation, on data sharing, and also that this is an area where we also see that transparency is a must, and the open science and open data, of course, keeping some metadata protected is also very important. We also have seen finally that these are also all kinds of big data that are coming to our plate to improve food safety, and this, of course, is also additional challenges that we have ahead of us. So thank you very much for being with us. Thank you to the technical colleagues that facilitated the video, the web streaming, and also my co-chair and the rapporteurs, and in particular thanks to the session coordinator, Winnie, and all the effort that all the colleagues in EFSA have been putting in having this successful session. Thank you very much. There are now poster pitches in the central area, so I'll invite you to be there, and I understand that the biological hazards posters will be today pitched. So good buon proseguimento, we say in Italian. The flies are really